Department of Analytical Biochemistry focuses on the analysis of biomolecules in complex biological samples such as blood, urine, or tissue from patients. We focus especially on the analysis of proteins with two main goals. One is the analysis of biomarkers, or the discovery of new biomarkers and the validation thereof. And the second part is the analysis of biopharmaceuticals. That means proteins that are used as pharmaceutical products. The department is quite international. We have about 10 nationalities of PhD students, postdocs, technicians working all in the area of chromatography and mass spectrometry. As an example, we started a multicentric effort together with the hospitals in Rotterdam and Nijmegen to discover and validate new biomarkers, which we call the Biomarker Development Center in the Netherlands. And one problem with biomarkers is shown up here, is especially that there's a lot of biomarker discovery, so there are thousands of new proposed biomarkers every year, but very few of these really make it to the actual clinical application. That means very few of these discovered biomarkers are useful in the end for the patient, and that's what they should be. And our goal is to reduce this gap and to develop methods and also approaches to improve the biomarker development pipeline. On the one hand, this gap is due to the design of the studies themselves. Many studies are fairly small, so we work immediately with larger numbers of samples from different centers. But the second gap is also that often the validation is not done properly. That means that methods are not really reliable and cannot be reproduced. And this is the focus of this group. We work on different areas, which you will see a little bit later. For example, we work on the improvement of the throughput of the analysis so we can analyze large numbers of samples. We try to improve the specificity of the analysis so we do not measure interferences, other molecules that are not related to this biomarker. And we try to improve the sensitivity of the method because many disease-specific biomarkers are present at very low concentrations in these complex matrices. In order to do so, we've established a consortium of private industrial partners, patient organizations, and academic labs, such as this. So this is what we call the Biomarker Development Center, the BDC. So how this works is that projects are fed in from academic groups, from research institutes, or from companies, and then we process them in a way you could say. We do the analyses and also the data analyses, and we prioritize the biomarkers based on concrete, robust data.
My name is Frank Kont. I'm a Dutch PhD student within this group, and I'm working on biomarker validation. In my project, we aim at validating promising COPD biomarker candidates in order to help lung physicians to diagnose COPD patients, as well as help them to optimize and personalize treatments. Specifically, I'm focusing on developing analytical methods using Multitoph mass spectrometry. Multitoph is a very fast technique, which is of course beneficial, but it also requires that the samples that you introduce to the mass spectrometer are very clean and highly enriched. This is the challenge that I'll be facing in my project. High throughput takes center stage in my project for both the sample preparation as well as the mass spectrometric detection, and the ultimate goal is to develop biomarker assays that allows the clinicians to help the patients, and therefore we hope to contribute to the well-being of the patients.
Hello, my name is Arangay, and I am originally from Spain. There I studied my degree of chemistry and also pharmacy, and did my PhD in analytical chemistry. Once I finalized my PhD, I moved here to Groningen, to the lab of Professor Bischoff, in order to learn a bit of liquid chromatography coupled to mass spectrometry, since it is a very interesting technique when it comes to the pharmaceutical industry, mainly for drug characterization, but also in my field of research, which is biomarker discovery and validation. And well, I've been working here for the last 4.5 years, and well, It's a very nice working environment. It's very multicultural. There's people coming from all over the world. That makes it personally quite interesting. But also when it comes to work, we have a very interdisciplinary environment. We have collaborations with groups from different types of expertise within the Netherlands and also outside the Netherlands. We collaborate with organic synthesis groups, with groups that do toxicology, pharmacology and targeting studies. And well, I'm mainly working in those type of projects which involve more type of disciplines, and today I will show you a bit about that. Well, as I mentioned to you, we have a collaboration with different groups, and I will present today one of those projects. And in this project, what we are mainly working is to develop new fixatives for tissue fixation, and that we do in collaboration with the Leiden University and also with the toxicology department here at pharmacy. And what we are basically doing is we get rat liver tissue that they make certain cores and then they slice them. So you can see here what type of small pieces of tissue we have. Then what we do is we fixate them with different fixatives, and that you can see here, it's just by placing them in a solution and leaving them overnight. And then what we can do is we dehydrate those tissues and we embed it in paraffin, and we get this type of paraffin blocks. And from these paraffin blocks, what we can do is to cut some sections and put them on microscope slides. So you can, by doing some staining, as you can see here, that it becomes colored, just see the morphology of the tissue. So once we see that our fixatives allow to preserve the tissue, now we want to see if they have an advantage for what we do, which is the analysis of proteins on tissue. And how we do that? Well, I've made here also kind of a scheme, so you can see a bit. Basically, we take our tissue that we have here, we have to break it in small pieces, and to do that, we usually use this type of thing, which is a mortar, and we crush the tissue into small pieces. Then what you do is you add a solution to this tube in order to extract the proteins. So you would end up with a solution in which all your proteins are dissolved. Then we cleave those proteins into small peptides by doing trypsin digestion, also in this tube. And then we put what is coming out from this tube into an LC-MS vial, and we bring it to our instrument. And if you follow me, I will show you how our instruments look. So once we have our samples ready, what we do is to place them in our instrument. So here's the auto sampler where you can place them, and then they will be ready for analysis. This is the liquid chromatography part, and this is the mass spectrometer. In this case, it is a Q-top, which is a very accurate system. So once your samples are here, then it comes the work behind the computer. And here, basically, you just generate a sequence where all these samples will be analyzed and run through a method. And as you can see here in the screen, what we get is a chromatogram with the separation of all the peaks that are present in the sample. And based on the signature fragmentation of those compounds, what we can do afterwards is to identify them and see which proteins are present in those samples. And all this information, we usually employ it for biomarker discovery or validation. So we compare samples coming from healthy individuals and diseased people to see if in those samples are differences between the proteins which are present. And those proteins which are different, we consider them to be potential biomarkers of that disease. Once we have identified a potential biomarker, the next step is the validation. And when is it validated, then it can move to the clinics, which is the final aim of all our research.
Hello, my name is Kees Bronsema. For the last 4 years I've been doing my PhD research here at the Department of Analytical Biochemistry. While doing so, I was employed by a commercial research laboratory where I spent the last at least 10 years doing method development and validation for small molecules in plasma for support of pharmacokinetics. due to their extensive experience with proteomics here at the Analytical Biochemistry Research Department, there was a collaboration between the company where I work and the university in order for the company to learn about how to analyze proteins. During my time here, I've looked at different aspects of protein quantification, and we've set up analytical methods to quantify proteins of different molecular sizes from different matrices, all with the purpose of validating them to apply them in pharmacokinetic studies, but for proteins, not for small molecules. This is a big change that we see in the contract research, that our clients are moving from analyzing from producing small molecules to producing protein biopharmaceuticals.
My name is Asselman. I'm a first year's master student here at the University of Groningen. I decided to do my first research project here at the Analytical Biochemistry Department. The project that I'm working on has an aim to develop a quantification method using LC-MS for a protein called TRIL. TRIL is a very important protein because it can function as a biopharmaceutical in cancer therapy as well as a biomarker for autoimmune diseases. This project, I have faced a lot of challenges involving the sensitivity of the method. This is due to the low endogenous concentration, which is around 500 picograms per ml. To reach this low level, the method that is developed uses two different enrichment steps. One on the protein level and one on the peptide level. For the enrichment on protein level, IMAC is used as an enrichment. This technique is based on the amount of histidines present on the protein surface. The more histidines, the more it is retained on the column. After digestion, strong cation exchange is used as an enrichment on the peptide level. During this technique, our peptide of interest is positively charged and is retained on the negatively charged column, while other peptides are not. For the elution, the pH is increased and our peptide becomes neutral and elutes easily from the column. After this, the sample can be injected on the LC-MS system.
I am Peter Horvatovic. I am working at the Department of Analytical Biochemistry as a bioinformatician, but I am analytical chemistry by main formation. I did my PhD in food analytical chemistry in Strasbourg in France, and after that I had occasion to work at pharma industry, and there I was working in molecular modeling and cheminformatics, having a lot of programming tasks, which I found very interesting, but I always wanted to combine analytical chemistry with informatics and bioinformatics and mathematical modeling. And I get occasion to develop that aspect here at University of Groningen in a group of Rainer Bischoff, focusing on the analysis of proteins and peptides in complex biological fluids, and to find biomarker to fish out the right compounds to detect a certain stage of disease, especially which are in the early stage, because then people can get better treatment and have more chance to be cured efficiently. And so I am combining there analytical chemistry and programming, cheminformatics, analysis of the large amount of data. I'm writing a program, for example, you can see here as a MATLAB script, or making a volcano plots. Those are plots that visualize all the compounds that are detected by a complex LC-MS analysis, and all the plots represent one compound. And we are only interesting for the compounds which or peptide which are very discriminative between healthy and disease stage. And those are the plots which are selected here in red, show large difference between healthy and disease stage, and I am fishing out that from this large amount of data. And here I can visualize the part of the chromatogram which contain those plots, and look if all the processing went right and if the statistic is good. in one color, either red or blue, represent data coming from healthy or disease stage, and we are looking further of what those peptides and the originating protein represent for the disease. So this is the starting steps for biomarker discovery, and of course we have much more processing than this is just one example. Now I have a large set of processing just ongoing on the cluster, so you can see all the process in a large cluster with half terabyte of RAM and 48 core of processor that we are using to process this large amount of data.
My name is Hjalmar Permentier. I am the head of the Interfaculty Mass Spectrometry Center. We provide mass spectrometry service for the University of Groningen and the UMCG, and we work for many different departments, including pharmacy, chemistry, biology, and more also the medical sciences from the University Medical Center. We provide service for all kinds of different applications, ranging from proteins, oligonucleotides to polymers, small molecules, including drugs and drug metabolites, both qualitatively and quantitatively. And the mass spectrometry facility, we now have 16 different instruments with which we can do a variety of different analyses. And in a year, the number of analyses is in several thousands for several dozen different research departments. We cover almost the entire science faculty in projects that we support over a year. The projects we support, the instrumentation that we have in the lab is mostly liquid chromatography mass spectrometry. In addition, we have several multi-TOF systems, and the type of mass analyzers ranges from triple quadrupole mass spectrometers for quantitative analysis to ion traps and quadruple time of flight and Orbitrap instruments, which are mostly used for high resolution analysis of proteomic samples.
I'm Annie van Dam and I'm part of the mass spectrometry core facility. This is the group where researchers from different groups within the university or the hospital come if they want support for their projects, if they want support with mass spectrometry. And it can be a question about small molecules or protein, and sometimes we have to identify the samples. It is going about modulation of protein or chemical reaction, but there are also quantitative questions, and it depends very much about the question how we handle the problem. Afternoon we are going to have an appointment with somebody of the group of Genie Groothuis. That is the Department of Pharmacokinetics, Toxicology and Targeting. And he has a question about analysis of quinidine and its metabolites in liver slices. It will be a quantitative project, and we have to be aware that the metabolites have the same molecular weight, so probably the HPLC separation is also very important.